This is a revision video for Unit 2 of AQA GCSE Biology or Combined Science. About 40% of the marks in the GCSE Science exams are for pure recall of facts from the specification. I've written these questions, which you can find in the description below, so you can check how well you've memorised these key facts before moving on to questions that ask you to apply your knowledge or to analyse data. You could download the questions and answer these before using the video to check your answers, or just use the video to help you make flashcards. A tissue is a group of cells that have a similar structure and function. For instance, your organs are made of muscular tissues which contract, glandular tissues which produce hormones and other chemicals, and epidermal tissues which cover the outside of an organ a bit like a skin. When these tissues are aggregated together, they can form an organ. Where lots of different organs work together to perform a specific function, this is called an organ system. One example of an organ system is the digestive system, and you should be able to label the mouth, the esophagus, the liver, the stomach, the small intestine, and the large intestine. You also need to know that just underneath the stomach is the pancreas. Besides enzymes, the glandular tissue in the stomach makes hydrochloric acid. The purpose of digestion is to break down large food molecules into smaller, soluble molecules which can be absorbed into the bloodstream. These products of digestion are used to build new carbohydrates, lipids, and proteins, while some of the glucose is used in respiration. An enzyme is a biological catalyst. In other words, it speeds up rates of reaction. It's made from protein and it has a specific active site that is complementary to its substrate. This means that the active site has the exact same shape as the substrate, and so they fit perfectly together. This is also known as lock and key theory. The optimum pH is the pH at which an enzyme works best or has the highest catalytic activity. Amylase is made in the mouth, the small intestine, and the pancreas, and it works to break down starch into simple sugars. Proteases are made in the stomach, the small intestine, and the pancreas, and they work on protein. When proteases break down protein, they make amino acids. Lipases are made in the small intestine and the pancreas, and they break down lipids, in other words fats and oils, and they make fatty acids and glycerol. The pH in the mouth is approximately neutral. It can vary between about 6.5 and 7.5, whereas the pH in the stomach is much lower due to the hydrochloric acid and can vary between about 1.5 and 3.5. The pH in the small intestine is generally ever so slightly alkaline. This is partly due to the role of bile. Bile is slightly alkaline and therefore can neutralise the pH from the stomach acid, and also it works to emulsify fats. This means that it breaks them down into small droplets which increases their surface area. Both the pH and the emulsification of the fats helps them to be digested much quicker than they would be otherwise. The first required practical in Unit 2 of GCSE Biology is the food test required practical, and you need to know the tests for sugars, for starch, for protein, and for fats. The test for simple sugars is called the Benedict's test. It involves using a chemical called Benedict's reagent, which is made out of copper sulfate with some sodium carbonate and sodium citrate in there. You add your sample to the Benedict's reagent and then you boil this. You could talk about heating it to a very high temperature, but it's definitely not enough to just say that you would warm it. We want this idea that it needs to be heated up very, very hot indeed. If sugars are present, then we'll start to see a colour change. The Benedict's reagent itself is blue because of the copper sulfate, but if there's sugar present, then it will turn green, yellow, orange, or even red depending on how much sugar is present. To test for starch, we use an orangey brown iodine solution, which will turn blue-black in the presence of starch. The test for protein is the Burette test. Be very, very careful of your spelling of this word. It's one of the very few examples in GCSE Science where if you don't spell it correctly, you won't get the mark, because it shows that you're confusing it with the piece of equipment that you use in chemistry for measuring volumes. In the Burette test, we use a strong base like sodium hydroxide or potassium hydroxide together with copper sulfate solution. And in the presence of peptide bonds, which are found in proteins, this will change from blue to purple. This is a really good test because it isn't fooled by amino acids on their own. You have to have entire strings of protein for it to work. There are lots of different ways that you can test for fats, and there isn't actually one named in the specification. But some of the different examples you might have met include the paper test, where you rub a fatty food on a piece of paper and look for a translucent stain around it. But obviously this doesn't work if you have a wet food, like milk for instance. You can also crush the food with water and filter it through and see if this separates out. Or you can crush the food with ethanol and then add water and see whether you get a white emulsion. In the second required practical of Unit 2 of GCSE Biology, you look for the optimum pH of amylase. To do this, you use a continuous sampling technique, and this means that you're taking samples at regular intervals, maybe 10 seconds or 30 seconds, and observing the changes that happen. You're not just waiting for the end of the test and then reaching a conclusion. During this test, you could maintain the temperature of the amylase starch mixture by using a water bath or an electric heater. It wouldn't be appropriate to use a Bunsen burner because you couldn't control the temperature finely enough. The control variables during this investigation will include the volume of the starch solution, the volume of the amylase solution, the concentration of the starch solution, and the concentration of the amylase solution. When you're carrying out the required practical, your first step will be to assemble several identical samples of your starch solution. This is important because the volume of the starch solution and the concentration of the starch solution are two of your control variables. Next, you need to add some pH buffer to each one of your samples, and this should be the same volume because again, this should be controlled. So for instance, you might have one set of starch at pH 1 and then another set at pH 4 and another set at pH 7 and so on and so forth. You need to put the samples and also your amylase enzyme into a water bath to reach a steady temperature, for instance 37 degrees if you're using human amylase. So we want them all to be at the same consistent temperature. Then you need to get your spotting tile ready, and you put small drops of iodine solution into every well. You then add a set volume of the amylase, maybe 1 centimetre cubed again, to your first starch sample and mix it really thoroughly so it's properly homogenised. At regular intervals, say 10 seconds or 30 seconds or 1 minute, you need to remove a sample from the test tube and pipette this into some of the iodine in the spotting tile. If there is still starch in the sample, then the iodine will turn blue-black. If all the starch in the sample has gone because it's been digested by the amylase, then it will stay orangey-brown. You continue doing this at regular intervals until the iodine doesn't change colour, and then you write down the time that took. Then you get your next tube of starch, which has a different pH, and you repeat the entire experiment. Whichever pH takes the least time to stop changing colour is the optimum pH out of the ones that you've tested. That might not be the exact optimum pH because there's a limit to how many different pHs you tested. So for instance, if you checked pH 4 and pH 7 and pH 10, you might know that 7 is the best out of those, but that doesn't actually tell you whether the optimum pH is 6 or 8, for instance. So in order to improve this investigation, you might want to repeat it using finer gradations of pH. In the respiratory system, you should be able to label the trachea with its cartilage rings to prevent you from having your windpipe crushed, the bronchus where the trachea splits in two, the bronchioles where each bronchus then splits into smaller branches, and the alveoli, which are those small cloud-like structures which increase the surface area of the lungs. The human lungs are adapted by having those alveoli, which increase the surface area, by having only a single cell thick in the walls of those alveoli, so the diffusion pathway for oxygen and carbon dioxide is minimised as much as possible, and also by having a very, very good blood supply because there are lots and lots of capillaries. On the heart, you should be able to label the right and left atria and ventricles. Remember that the heart is labelled from the point of view of the person whose heart it is, so when you're looking at a piece of paper, right and left are not necessarily where you expect them to be. You should also be able to label the major blood vessels, the pulmonary artery and vein, and the aorta and vena cava. You should also know that there are valves inside the heart in between the atria and the ventricles, preventing backflow of blood. When we talk about the heart having a double circulatory system, this is the fact that the heart is split in two. So the right ventricle pumps blood to the lungs where gas exchange takes place, and then the left ventricle pumps blood around the rest of the body, which is why it's much more muscular and just generally stronger because it's got to pump the blood further. The blood leaves the left ventricle via the aorta and it returns via the vena cava into the right side of the heart. Then the blood leaves the right ventricle via the pulmonary artery to go to the lungs and it returns via the pulmonary vein after it's been oxygenated. Connecting the heart to the lungs are the pulmonary vein and the pulmonary artery. The major blood vessels connecting the heart to the rest of the body are the vena cava and the aorta. The pacemaker is a small group of cells bound in the right atrium which control the body's resting heart rate. If you have an irregular heart rate, what we call an arrhythmia, then this can be corrected with an artificial pacemaker, which is an electrical device. The three types of blood vessels are the arteries, the veins, and the capillaries. Now, the arteries take blood away from the heart. So because it's under high pressure, they have a thicker layer of muscle and they have a thicker layer of elastic tissue, because this allows the blood vessel to sort of expand because of that high blood pressure. The veins, which carry blood towards the heart, have thinner walls, less elastic tissue, but they also have a wider lumen, which is the hole in the centre. They also have valves, which help to prevent backflow. The capillaries are a single cell thick, and this is to allow the rapid diffusion of gases to and from tissues. In order to calculate the blood flow rate through a blood vessel, you need to know the volume of blood that has passed through that vessel, and that could be given to you in centimetres cubed or decimeters cubed, and you need to be able to convert between them. And then you should divide this by the amount of time taken for that blood to flow. The plasma is the liquid part of the blood, and dissolved in this you find glucose, amino acids, carbon dioxide, lactic acid, and also floating in there, but not dissolved of course, are blood cells and hormones. The red blood cells are responsible for transporting oxygen in the form of oxyhemoglobin. The white blood cells are part of the immune system, and they're responsible for killing or destroying microorganisms through phagocytosis, antibody production, and also antitoxin production. The platelets are responsible for helping the blood to clot. Red blood cells are adapted to carry lots of oxygen in that they're full of hemoglobin, which can bond to the oxygen. They have a biconcave shape to increase their surface area, and they also have no nucleus to make even more room for hemoglobin. A non-communicable disease is one that cannot be transmitted between people because it isn't caused by a pathogen. Fatty material like cholesterol, sometimes called plaque, builds up, and this narrows the arteries and slows down the rate of blood flow through them. This means that less oxygen gets to the heart muscle, which can lead to a heart attack. Using drugs such as statins avoids the need for surgery, which can be expensive and dangerous, and it's a long-term change. As long as you're taking those statins, then the cholesterol that's in your diet will have less of an impact on your heart. In terms of disadvantages, you could have an adverse reaction to them, so there could be side effects. The person who's taking them needs to actually remember to take them, and also it won't immediately solve a long-term build-up of cholesterol. So if there's already a problem and that problem is looking pretty urgent, then the drugs alone aren't going to fix it overnight. In terms of mechanical solutions, like using stents, they're more immediate, but they do require surgery, which, as we've said, can be expensive or dangerous. And also, just because you've cleared out the cholesterol that's in one artery doesn't mean that it couldn't then build up in other arteries. Having a heart transplant is kind of an immediate solution in that it's giving you an entirely new heart. So whatever the old problem is, it's out of the way. And in some instances, that may be the only available solution because the heart is so badly damaged. But again, it requires surgery, which is expensive and dangerous, and there's a risk of rejection if the organ is not closely enough aligned with the person who's receiving it, and also the person who's receiving it is going to need to take immunosuppressant drugs for the rest of their life, and this means that they're more susceptible to catching other illnesses. A stent is a wire cage that's placed into an artery in order to keep it open. People's valves could become faulty if they stop fully opening or if they start leaking, and this can lead to heart failure, strokes, and blood clots. Two instances in which we might use an artificial heart are to keep patients alive while waiting for a heart transplant, or to allow the heart to rest as an aid to recovery. AQA define health as the state of physical and mental well-being. In addition to communicable and non-communicable diseases, your health can also be affected by diet, stress, and life situations. There are probably lots of other things as well, but they're the three that are named in the specification. Having a defect in your immune system can make you more susceptible to infectious diseases. Certain viruses, like HPV, can act as a trigger for certain cancers, like cervical cancer. Some people, when they have an immune response to an infectious disease, can then have allergies triggered, and this could lead to skin rashes or asthma. Having poor physical health can also have an impact on your mental health. A risk factor is an aspect of your lifestyle or substances that are in your body or environment that are associated with an increased risk of disease. Now, this could be something that's causative. So, for instance, we know that there's a link between the chemicals in cigarettes and lung cancer. Or it could just be something that is correlated with it, but we don't know why. Diet, smoking, and exercise are all known risk factors of cardiovascular disease. Obesity has a strong link with the development of type 2 diabetes. Regular consumption of alcohol has a known impact on liver and brain function. Smoking, of course, has a causative link with lung disease and also lung cancer. Both smoking and alcohol have an impact on the health of growing unborn babies, and carcinogens, in other words, chemicals that cause cancer or ionising radiation, are also risk factors in the development of cancers. Cancer is the uncontrolled growth and division of cells as a result of a series of mutations. A benign tumour is a group of normal cells that are growing and dividing in one area, and they're usually contained within a membrane, but they don't invade other parts of the body. They can still be dangerous because they can still crush other organs and make you unwell in other ways, but they're not necessarily moving around the body. A malignant tumour is a cancer that invades neighbouring tissues and can spread to different parts of the body in the blood, where they form secondary tumours. On a diagram of the leaf, you should be able to identify the waxy cuticle, the upper epidermis, the palisade mesophyll, the spongy mesophyll, the vascular bundle consisting of the xylem and phloem, the lower epidermis, and then the guard cells which sit on either side of the stoma. The waxy cuticle reduces water loss. The epidermal layer is basically transparent. It contains very few subcellular structures, and that allows the maximum amount of light to get through to the palisade mesophyll, which is where most photosynthesis is going to happen. Those palisade cells are packed full of chloroplasts in order to absorb the light. The spongy mesophyll has all of those gas spaces to allow carbon dioxide and oxygen to diffuse through the leaf. The two vessels in the vascular bundle are the xylem and the phloem. The pores in the surface of the leaf are called stomata, and these pores are there to allow gas exchange. The cells on either side that open and close them are called the guard cells. The xylem are hollow tubes strengthened by lignin, and there is no barrier in between the end of one cell and the start of the next one, so this allows water to just move through the xylem unimpeded. The phloem are tubes of elongated cells, and they do have cell walls at the end of each cell, but there are pores in these, and so this allows sugars and other materials to flow through the cells. The root hair cells don't contain any chloroplasts because they're underground, so they can't do any photosynthesis, and they also have a very large surface area to maximise the amount of water and also nutrients like nitrates and magnesium that they can absorb. The movement of water through the plants from the roots up to the leaves and out is called transpiration, and it takes place through the xylem. As the temperature increases, so does the rate of transpiration, because those water molecules have more energy and therefore they move faster and therefore they diffuse faster out of the leaves. As humidity increases, transpiration decreases. This is because the concentration gradient between the water in the leaf and the water in the air is reduced. It becomes shallower and less steep, and therefore diffusion happens more slowly. Under high light conditions, the plant will do more photosynthesis, and in order to do that, it needs the reactants, in other words, water and carbon dioxide. To get more carbon dioxide, it has to have its stomata wide open. So high light conditions lead to more transpiration. Having high air movement, for instance on a very windy day, will also increase the rate of transpiration. This comes back again to the concentration gradient. The windier it is, the more the water that has just exited the plant leaves will be moved away from the plant, and this helps to maintain that concentration gradient. If there isn't any air movement, then the water that's just come out of the leaves stays there and basically makes the air next to the plant very humid, and that would reduce the rate of transpiration. Translocation is an active process. In other words, it takes a lot of energy. It moves assimilates, sugars, from where they're made in the leaves to anywhere in the plant that they're used, so particularly in storage organs like tubers. It can move sugars about 10,000 times faster than diffusion alone would do. This takes place through the phloem. Thank you very much for watching. Hopefully you're now feeling more confident that you can recall the facts from the specification for GCSE Biology Unit 2. If you did find the video useful, then don't forget to like and subscribe and share. There'll be more GCSE Biology videos coming soon.